Hi friends, today we'll be discussing about COVID-19. Okay, because this is the most important topic right now, and I believe that there will be many questions appearing in your PG exams, and it will also give you an insight about COVID-19. Now, to start with the basics, let's start with the virology. You need to remember that COVID-19 is nothing but a disease. It is not the virus. Okay. So COVID-19 is a disease that is caused by SARS CoV-2 virus. Okay. So COVID-19, the full form of COVID-19 is nothing but coronavirus disease which occurred in the year 2019. Okay. Now this SARS CoV-2 virus is basically a single stranded positive RNA virus. Okay, it is of the beta coronavirus group. We'll be seeing the groups further. Okay, and it infects the host receptors, mainly the receptors which are present on the endothelial cells of, for example, on the alveoli, that is the ACE2 receptors. Okay, it can also infect the attached to the receptors, that is the cellular protease TMPRSS2. Okay, the right now there are mainly various types of variants, of which the most important ones are G614 variant and the D614 variant. The International Committee of Taxonomy of Viruses has named the virus causing COVID-19 disease as SARS-CoV-2, that is severe acute respiratory syndrome coronavirus 2. Okay, the taxonomy of viruses consists of a 15 rank structure for your information point of view, which starts with the realm, then further divided into kingdom, further phylum, then the class, order, then family, genus, and the species. Now, SARS-CoV-2 is basically the species which is of the genus beta coronavirus, the subfamily of which orthocoronavirinae, then of order Nidovirale. Now, the SARS-CoV-2 mainly consists of four major proteins, that is the nucleocapsid proteins, the envelope proteins, the spike glycoprotein, and the membrane protein, which we'll be seeing and discussing in the further slides. Let's see the structure of the coronavirus. Now coronavirus, as we all know, is basically spherical in shape, and it is of the diameter of approximately 125 nanometers. Okay, it is made up of basically four major structural proteins. Now we'll discuss these four major structural proteins one by one. Okay, starting with, let's discuss first which are these four major structural proteins. As you can see in the slide, the major structural proteins, four major structural proteins, is the S protein, then the M protein, then the E protein, and then the N protein. Now we'll see one by one what are these proteins and what is their role. Okay, now starting with the S protein, or the starting with the S protein, also known as the spike protein. Okay, the spike protein is a club shaped protein. Okay, you can see this club shape. Okay, the club shaped protein, and giving the coronavirus a crown like appearance. So the spike proteins gives the coronavirus a crown like appearance, giving it its name, that is corona, or the crown like appearing virus. Now this S protein plays a major role in attachment to the host membrane. Now when it attaches to the host membrane, okay, it is cleaved by host cell proteins. Mainly these are furin like proteins. Okay, when the S protein attaches to the host membrane, it is cleaved by furin like proteins, basically into two components. One is S1 and another is S2. Now this S1 is the major receptor binding domain. Okay, and S2 is the stalk of the spike protein. Okay, so this is the S1 domain, and then this is the stalk of the S2 component of the spike protein. The M, or the M or the membrane protein. Okay, so the membrane protein is, you need to remember the most important point, M for it is the membrane protein, and it is the most abundant protein in the four major proteins. Okay, now the function of this M protein, which you can see here, this is the M protein. Okay, this is the M protein. Now this M protein plays a major role in assembly of viruses. We'll see how it plays a role when we'll be seeing about the coronavirus entry into the host and its translation and transcription process. Next comes the E protein. The E protein plays a major role in assembly and release of virions which are produced within the host cell. Okay, and finally we are coming to the N protein. It is also known as the nucleocapsid protein, which plays a major role in RNA, viral RNA synthesis. Okay, so it plays a major role in viral RNA synthesis. Apart from these four major proteins, there is another protein which is known as the HE protein, also known as the heme agglutinin esterase protein. Now this heme agglutinin esterase protein plays a major role in adsorption of virus to the membrane host. Okay, so these are the major proteins which you need to remember. This is the slide which we have already discussed, that is the coronavirus composition. It is basically a medium sized RNA virus. Okay, it is one of the largest known viral RNA. It has the largest known viral RNA genome. Okay, next there are five structural proteins which are S or the spike proteins. Then there is the membrane protein which plays a role in viral assembly, nucleocapsid protein which plays a role in viral RNA synthesis, then there is the HE glycoprotein, and then there is E protein. Apart from this, you can remember here that the HE glycoprotein is basically seen in beta coronavirus genesis. Okay, and which is SARS CoV-2. As we know, we have already seen SARS CoV-2 belongs to the beta coronavirus genus. Now let's see the process of entry of the virus into the host, then the process of replication, transcription, translation in the next few slides. As we know, coronavirus basically reaches the lungs via respiratory droplets. Okay, on reaching the lungs, then it passes through the trachea, bronchus, bronchioles, reaching the alveoli. Now first let's see the basic structure of the alveoli. The alveoli is basically made up of type 1 pneumocytes which play a major role in gas exchange. That is, they play a major role in intake of oxygen and to release carbon dioxide. Then there are type 2 pneumocytes which play a major role in formation of surfactant. Then there are a few macrophages. As we had seen in the first slide, the receptor for coronavirus is the ACE2 receptors. These ACE2 receptors are present mainly on the type 2 pneumocytes. Now, before we go to the next slide, we need to remember here that angiotensin 1 is converted to angiotensin 2 by angiotensin converting enzyme. This plays a major role in increasing the BP. Whereas angiotensin 2 is converted to AT, angiotensin 127 by angiotensin converting enzyme 2. This angiotensin 127 plays a major role in decreasing the BP. So the mechanism of ACE is different from that of ACE2. Now consider this to be type 2 pneumocyte, okay, with ACE2 receptors. Now what happens is the spike protein which is present on the coronavirus attaches to the ACE2 receptors. Okay, so the first step in entry of the coronavirus into the cell is attachment, which occurs by the help of the spike protein or the S protein into to the ACE2 receptors. Once there is attachment, there is adsorption of the coronavirus into the host membrane. Okay, we have seen this previously. This adsorption is done by which protein? That is heme agglutinase esterase protein. Okay, once there is adsorption, next what occurs is the coronavirus comes inside the host cell. Once it is inside the host cell, there is uncoating of the virus. Okay, once there is uncoating of the viruses, there occurs genomic replication, and this genomic replication basically occurs into the nucleus, giving rise to the messenger RNA. Okay, so this is a messenger RNA. Okay, this is a single stranded messenger RNA. Now this messenger RNA, with the help of ribosomal RNA, undergoes the process of translation outside the nucleus to give rise to proteins. Which proteins? These are the major structural proteins, that is S, M, N and E proteins. Okay, these proteins are then assembled. As we have seen previously, the assembling role is by the membrane proteins and also by the E proteins. Okay, so they are assembled to form virions. Okay, so they form the virionic structures, okay, with spike proteins, and these virions are finally released. Okay, so assembly is mainly done by membrane proteins. Then these are released into the outside the host cell, and the release role is mainly played by the E proteins. Okay, so this is how the process of entry of coronavirus into the host cell occurs. Basically, there is attachment of the coronavirus with the help of spike proteins to the ACE2 receptors. The ACE2 receptors, then there is adsorption within the host cell, then there is uncoating of the coronavirus, and there is formation of the uncoating leading to formation of the mRNA, where there can occur replication, and with the help of ribosomal RNA there occurs translation. Translation is basically nothing but the process of synthesis of proteins. Now the various proteins which play are synthesized are mainly the structural proteins and all the associated required proteins, that is S, M, N and E. Now these S, M, N, E proteins, they are assembled by the membrane M membrane protein mainly, and then they are released outside the host cell into by with the help of the E protein. Okay, once the virions are released out of the newly formed virions, when they are released out of the type 2 pneumocytes, okay, there are large number of virions getting released. Okay, sometimes a part or component of the virions, like only the spike proteins, nucleocapsid proteins, are released into the alveoli. This leads to the movement of the inflammatory cells within the alveoli, like neutrophils, macrophages, etc. And these inflammatory cells cause release of pro-inflammatory cytokines like interleukin 1, interleukin 6, tumor necrosis factor. These cytokines lead to vasodilation, okay, and finally increasing the permeability of the blood vessels so that more fluid enters the alveoli, leading to alveolar edema. So these alveolar edema, so these cytokines basically sometimes cause is release is so much, leading to what is known as the cytokine storm syndrome. Okay, and as there is increase in the inflammatory cytokines like interleukin 6, okay, we use some anti interleukin 6 inhibitors like tocilizumab in the treatment of coronavirus or the cytokine storm syndrome, which is caused by the coronavirus. This is basically the revision of the previous slides which we have seen, the virus basically attaches to the host cell. Once there is a attachment of the whole coronavirus to the host cells with the help of ACE2 receptors, there is adsorption of the virus into the host cytoplasm, after which there is uncoating of the virus. Then inside the nucleus there occurs the genomic replication, giving rise to formation of the mRNA or the messenger RNAs. Then these, with the help of the ribosomal RNA, undergo a process of translation leading to formation of new proteins, and these proteins are the major proteins. These proteins then assemble to form the virions. Okay, once we are well versed with this, we can also see that what are the various researches that are going on to prevent this entry of virus into the host cell. Okay, so basically vaccine is from can be produced from this spike protein. Okay, then there is research for fusion inhibitors. Okay, so that there is no fusion between the host receptor and that of the cell. Then there are drugs in under research for which play a role in receptor blockade. Finally, there can be research among about protease inhibitor drugs. Okay, here I would like to tell you about a drug called as Remdesivir. Okay, so remember, Remdesivir is nothing but an adenine analog. Okay, now before we discuss how it works, if you see the virus on entering the human cell, this virus on entering the human cell, there is uncoating, and this uncoating really leads to release of the RNA. Okay, so this uncoating leads to release of the RNA. This RNA, in presence of RNA dependent RNA polymerase, okay, will lead to formation of new RNA components. Right? Now these RNA components are basically made up of adenine, guanine, cytosine, uracil nucleotides. Okay, this is how, and these newly formed RNA components play a role in the formation of virions. Okay, now since remdesivir is an adenine analog, when the remdesivir enters the human cell, it gets converted to its active form. The active form is known as GS441524. Okay, which is an adenosine triphosphate analog. Now, since it is an adenosine triphosphate analog, the RNA dependent RNA polymerase wrongly recognizes this GS441524 as an adenine nucleotide, and instead of inserting adenine into the RNA component of the new virion, it inserts the remdesivir component, and this leads to termination of the process of synthesis of new RNAs, and hence new virions are not produced, and this is how remdesivir works.